Hello everyone. I welcome you all to Biology Lessons with Kalyani. Today we will be talking about a new topic, that is carbohydrates. So this is a snapshot of the CBSE textbook. These are the important points which we will be covering today. So let us first see what are carbohydrates. Carbohydrates are polyhydroxyaldehydes or ketones. It means they are a class of biomolecules which have many hydroxyl groups in them, and they can either have an aldehyde group or a ketone group in them. If the carbohydrates are having an aldehyde group in them, they are called as aldose. If the carbohydrate is having a ketone group in them, they are called as ketose. So example of aldose is glucose because it is having an aldehyde group in them. An example of ketose is fructose, which is having a ketone group in them. Right? Now these carbohydrates can contain carbon, hydrogen and oxygen. Sometimes they can also have nitrogen, phosphorus or sulfur. Let us now see how we can classify carbohydrates. Carbohydrates on the basis of number of sugar units present in them can be classified as monosaccharide, oligosaccharide and polysaccharide. Here saccharide is derived from the Greek word saccharon, which means sugar. Now monosaccharides typically consist of 1 sugar unit, oligosaccharide contain 2 to 10 sugar units, and polysaccharides, they contain more than 20 sugar units in them. Now these sugar units are attached to each other by a special kind of bond. It is called as glycosidic bond. Now if we see examples of monosaccharides, it is glucose, fructose and galactose. If we see oligosaccharide, it will have disaccharide which has 2 saccharide units in them. Example are sucrose, lactose, maltose. Then it can also have trisaccharide which has 3 sugar units in them. Example can be raffinose. Now also 4 saccharides can be present, which is example is stachyose, and the type of saccharide is called as tetrasaccharide. If we see polysaccharides, examples of them can be starch, cellulose and glycogen. So here only disaccharide is important, monosaccharide you can see and polysaccharides. You can just remember trisaccharide and tetrasaccharide example. Let's see hydrolysis of saccharides. Now I told monosaccharide consists of only 1 saccharide unit. So if you hydrolyze monosaccharides, it cannot be further hydrolyzed into any simpler residues, right? But if you hydrolyze oligosaccharide, then let's say it is a disaccharide, you hydrolyze it, so you will obtain 2 saccharide units. Similarly, trisaccharide if you hydrolyze, 3 units will be obtained. Tetrasaccharide if you hydrolyze, 4 saccharide units will be obtained on hydrolysis. Now if we consider polysaccharide, multiple units which are constituting the polysaccharides will be obtained. Clear? Let's see in detail monosaccharide. Now monosaccharide are simplest sugar. We already discussed they cannot be hydrolyzed to any simpler carbohydrates. Now they are represented by the formula CnH2nOn. Here N ranges from 3 to 7. Typically these monosaccharides are soluble in water, sweet to taste and generally they act as a source of energy in the cell. If we see examples of monosaccharides, then based on the number of carbon atoms, it can be triose. Triose means the single sugar unit will have 3 carbon atoms. Examples are glyceraldehyde and dihydroxyacetone. Similarly, tetrose will have 4 carbon atoms. Pentose will have 5 carbon atoms. Hexose will have 6 carbon atoms and heptose will have 7 carbon atoms. Examples of tetrose are erythrose and erythrulose. Pentose example are ribose and ribulose. Hexose is glucose and galactose and fructose. Heptose example is glucoheptose and pseudoheptulose. Right? So important point is that based on the number of carbon atom, what are the sugar types you are naming them, right? And typically pentose and hexose is very important. Now let's see disaccharide. Now disaccharide, as the name suggests, consists of 2 sugar units. Now if you hydrolyze the disaccharides, you will obtain 2 monosaccharide units. Examples of disaccharides are maltose, lactose and sucrose. Now maltose is found in germinating grains such as barley. That's why maltose is also called as malt sugar. Lactose is found in milk and hence called as milk sugar. And the common sugar we eat is sucrose, otherwise called as cane sugar or table sugar. Now sucrose is formed only by the plants. Right? After remembering this, let's see the glycosidic linkage. Now I told between the sugar units, special type of bond or the glycosidic linkage will be formed. Now in case of maltose, C1 of the first glucose unit combines with the 4th carbon of second glucose unit. Similarly, in case of lactose, C1 of galactose combines with C4 of glucose. And if you see sucrose, C1 of glucose will combine with C2 of fructose. Now here you are also seeing alpha and beta. Alpha and beta refer to the configuration of the C1 anomeric carbon. So here what happens, if the OH group with respect to the with respect to the 6th carbon, if it is pointed upwards, then we call it beta configuration. If it is pointing downwards, we will call it alpha configuration. So we'll read more about it in structural details. As of now, don't worry much about it. Next, let's see polysaccharides. Polysaccharides are long chain of sugar, so they have more than 20 saccharide units joined together. Now, these polysaccharides are typically not sweet, insoluble in water and hydrophobic in nature. Their molecular weight ranges from medium to high. Now, polysaccharides can be classified as homopolysaccharide, which consist of single or same type of repeating units. Alternatively, they can be heteropolysaccharide, which contain 2 or more different kinds of repeating units. Right? Pink is one type, blue is another type. Now, these homopolysaccharide and heteropolysaccharide both can be unbranched or branched. Let's see some examples of homopolysaccharide. First example is cellulose. Now we all know plant cells, they are made up of cellulose, paper and cotton fiber, that's also made of cellulose. Now cellulose is long and linear chain and it is made up of typically glucose which are linked together by beta 1,4 glycosidic bond. Similarly, if we see starch is also made up of glucose, but it consists of two types of structures. One is amylose, second is amylopectin. Amylose constitutes about 10 to 30% and amylopectin constitutes 70 to 90%. So what's the difference between the two structures? Amylose is a straight chain structure. It consists of glucose joined together in only alpha 1,4 linkage. If we see amylopectin, then in addition to alpha 1,4 linkage, it also has branching. Now, what is the meaning of branching? Branching means another glucose molecule will attach to the parent structure. Now, this attachment or this glycosidic bond is formed between C1 of one glucose unit with C6 of another glucose unit, and this branching takes place typically after 25th or 30th residue. Clear? So because of this type of structure, starch possesses a helical secondary structure. And within this helix, it can trap the iodine molecules, and when you test or add iodine drops, starch forms a blue color. Clear? Now, third example is glycogen. Glycogen is present in animals. It is found in liver and skeletal muscle. Its repeating unit is also glucose. Now, here the structure of glycogen is similar to amylopectin. But only difference is that in case of amylopectin, the branching is happening after 25th to 30th residue. But here it will happen early or quicker, after every 8 to 12 residues. Clear? The fourth example is inulin. Now, inulin is made up of fructose as a repeating unit. It is the storage polysaccharide of roots and tubers of dahlia and artichoke. Now, it is not digested by humans. It is prebiotic and it is used in kidney function analysis tests. Now, coming to glycosidic linkage, it has beta 1,2 glycosidic bond in it, right? Now, let's see heteropolysaccharide. Heteropolysaccharide means the repeating units in such polysaccharide are different, right? So, examples of them can be hyaluronic acid, heparin, chondroitin 4-sulfate and gamma globulin. So we all know hyaluronic acid is found in connective tissue, heparin is an important anticoagulant, chondroitin 4-sulfate is found in cartilage, and gamma globulin, we know that it is responsible for immunity, right? So here you can just remember the examples. If you can remember, then remember the repeating units. Otherwise, example is also okay. So till now we saw homopolysaccharide, which consists of single or one type of repeating units. If more than one type of repeating units are present, it is called as heteropolysaccharide. Now, there is third type of polysaccharide which is present, which is called as complex polysaccharide. What is complex polysaccharide? So, in this case, the repeating unit will be an amino sugar, which is nothing but chemically modified sugar. Now, example is chitin. Now, in case of chitin, the repeating unit is NAG or N-acetyl glucosamine. Now, many units of N-acetyl glucosamine join together. That's why chitin is an example of homopolymer. Right? Now, this chitin is found in cell walls of fungi and exoskeleton of arthropods. Is that clear? So, all these examples we have to remember. In today's class, we covered a general overview of carbohydrates. So, this should be the base on which you can build up the further concepts. First, remember this part. After that, you do the structure of monosaccharide, learn the structure of polysaccharide, and then finally you can have a grip over the subject. I hope you enjoyed the session today. Hit the like button and please share and subscribe. Thank you everyone. Have a wonderful day.